Welcome to the continuation of our lecture on protists. Today we're going to be talking about autotrophic protists. Now remember, autotrophs are different from heterotrophs because autotrophs make their own energy. Well, they actually use energy in their environment, abiotic energy sources, to drive their metabolic system. What does that mean? Well, that's things like sunlight. Sunlight is an available energy source that's abiotic. Sunlight's not alive. And autotrophs can use that energy source to drive their system. Similarly, some autotrophs can use chemical energy to do that. In fact, some of the first organisms on Earth may have been autotrophs that use chemical energy to drive their metabolic systems. Well, today a lot of the autotrophs we're talking about are actually photosynthetic organisms. And some of these organisms you might have thought were plants, but they're not plants. They're actually protists. Now, the protists we've discussed are generally single cells to this point, but now we're going to start talking about a group of protists that includes macroorganisms. So organisms that are formed by a collection of cells and make up massive living things. What do I mean by massive? Well, one example is giant kelp off the coast of California. This is a protist. It's an algae, and it's also a macroorganism that grows 100 feet tall. So that's a pretty big organism, and it's also very important in its ecosystem, both structurally and functionally. Because of that, we're also going to be talking about a lot of different animals that interact with that macroorganism because it's a bit of a keystone species in its system. So from here on out, our conversation is going to bring in a lot of different organisms, and we're going to start to move from an organismal level in taxonomy and start to talk about community level dynamics. Previously, our lectures included my head on the side as a little like talking face in the corner. We're going to try for a few lectures me just narrating the slides as I pass through them, and maybe that'll move things along quicker, and we can include some other content along with those slides. So, enjoy our discussion on autotrophic protists. Before we discuss individual kinds of autotrophic protists, I'll start by saying that one of the interesting characteristics of a lot of these organisms is how their chromosomes change number through their life cycle. Now, remember, with humans, we are diploid organisms most of the time. We have two sets of chromosomes, one from our paternal side and one from our maternal side, and we only are haploid in the form of gametes, so sperm or egg. Those are the only conditions that we humans are haploid. Well, that's not the case with all protists, and they have some interesting life cycles and life history cycles that show the kind of diversity in nature in regards to the number of chromosomes an organism can have. So remember what a gamete is. A gamete is a mature haploid male or female germ cell that is able to unite with another of the opposite sex in sexual reproduction to form a zygote. So in our case, a gamete is a sperm cell or an egg cell. A zygote is a diploid cell that results from the fusion of two haploid gametes. A zygote can also be thought of as a fertilized ovum. When the sperm cell, which is haploid, encounters the egg cell, which is also haploid, they combine to form a diploid zygote, a 2N zygote. That is a cell that has two sets of genetic information, one from the paternal side and one from the maternal side. In the case of things like plants, of course, that gamete is in the form of pollen, which could be analogous to a sperm cell. And the egg, well, we still call that an egg. But just like with us, the pollen and egg are both single reproductive cells which have half of the genetic information. And they are produced and they combined, and that's pretty much the limit of how they are involved in the life cycle of plants. So the process that we undergo is called gametic meiosis. You can see here we've split this example organism into 2N and N stages. On the bottom, you have diploid stages, which are 2N. That is, they have the chromosome information from both maternal and paternal sides. Above that line in the center, we have N, that is haploid cells, and in this case, those are gametes, and those have half of the number of genetic information. If you follow from the individual, you can see that through meiosis, the 2N state is reduced to N. That is, the number of chromosomes is halved, and those gametes are N. Or you could think of N as 1N because they're half the number of chromosomes. Those N, or haploid gametes, then recombine through fertilization and create a zygote. In our case, most of our life cycle is spent as this 2N individual. That is, after fertilization, we continue growing, we develop, we mature, we reproduce, we take care of our offspring. What we think of as life is in this 2N state, at least for us. Well, not all organisms work like that, and when we talk about autotrophic protists, we also have to talk about the diversity of different life cycles. What is a spore? You've probably heard the word spore before, and it's used a lot to describe reproductive cells, but when we talk about spores, let's make a distinction. So a spore is a minute, typically one-celled reproductive unit capable of giving rise to a new individual without sexual fusion, characteristic of lower plants, including the bryophytes and mosses, fungi, and protozoans. So how is a spore different than a sperm cell or an egg cell, that is a gamete? How is a spore different from a gamete? Well, the difference is that a spore, like a gamete, is haploid, but unlike a gamete, a spore can reproduce its numbers of cells, even in that haploid state. So just because it has half of the genetic information doesn't mean it can't reproduce and create a multicellular but still 1N tissue. That might be kind of hard to think about, but if you were to make an analogy to the human life cycle, that would be as if an egg cell could reproduce many egg cells that are still unfertilized, they're still 1N tissues, but they could continue to reproduce and make a multicellular mass of egg cell tissue. Now, if you were to further extend that analogy, some organisms would be able to live in that state externally or for a period of time. So a spore is like a gamete in that it's haploid, but unlike a gamete, a spore can then continue to reproduce its tissues and make a 1N or haploid individual. So let's look at a diagram to clear up what that means. Here's a pretty complicated life cycle, but it is very interesting once you understand the relationship between these haploid and diploid tissues. Let's start with the far right bottom organism, and that's a sporothallus, and that's 2N. So this sporothallus is a structure for this organism that's composed of 2N tissue. That is, it has chromosomes, it has genetic information from the maternal and paternal side. Well, there in those brown collections of cells, you see that there's a label there that says sporangium. Now, you might not know what sporangium is or what the word angium means, but you know what a spore is. And I'll tell you this, sporangium is a tissue where spores are created. So this is where, through meiosis, these 2N cells are giving rise to 1N cells. And you can see those leaving just below that. Those myospores, those are spores created through meiosis, are 1N. So now we've gone from a 2N tissue, the sporothallus, to a 1N tissue, the myospore. Well, those myospores will then swim freely in this aquatic environment and then establish somewhere. And once they settle down and they start establishing, they have not gone through fertilization, but they will continue to double and grow and metabolize. So this is an independent structure, an independent tissue, a life stage, and kind of like an individual organism that is reproducing its numbers but not increasing its genetic information. And we call that a gametothallus. This gametothallus is 1N. All the tissues and cells in it are 1N, but it continues to grow and reproduce, and it gets bigger, and finally it kind of looks like the sporothallus, but the difference is that its genetic information is halved. It's 1N. So this whole gametothallus is haploid. Then it goes through mitosis and modifies some of the structure of its cells and creates gametes. Now, what's the difference between those gametes and those myospores? Just like we just previously defined, we know that gametes are destined to be fertilized. Gametes contribute to fertilization. They don't continue to grow. So those gametes are going to leave the gametothallus and combine and fertilize and go from a 1N and 1N gamete to a 2N zygote. That zygote is now diploid, and that young sporophyte, that young zygote, will reproduce its numbers and eventually form a mature sporothallus. Now, this is a bit different than a lot of the life cycles you might be used to, because in humans, we don't go through all these different stages. We kind of cut it short. But there's a lot of protists that do have these stages. So let's look at a kelp that you might have seen before, Laminaria. Laminaria is, in its larger macro form, a sporophyte. That is, it's 2N. Within the Laminaria sporophytes, which is on the very left side, you can see a little subset of tissue. And what that's illustrating is that within that blade, that kind of leaf-like structure, there's these collections of cells called sporangia. And sporangia, again, are sites where spores are made through meiosis. Now remember, spores are not gametes because they are destined to reproduce as 1N tissues, while gametes are destined to fertilize each other. Well, these zoospores that have been created in the sporangia leave, and they stay 1N. They don't fertilize. There's no fertilization happening with these 1N tissues. They then reproduce and make gametophytes. Remember, gametophytes are 1N tissues, multicellular 1N tissues. Those gametophytes then undergo mitosis and create specialized 1N tissues, which are gametes. And you can see that on the far right side. They've created an egg and a sperm. Now, going from a gametophyte to an egg or a gametophyte to a sperm, there's no reduction in the number of chromosomes because that already happened back when the sporangia made the zoospores. Those egg and sperm will then leave, and remember, since those are gametes, they will combine through fertilization. And once they fertilize each other, you then have a 2N zygote. That zygote will reproduce its 2N cells and create a sporophyte. And then that sporophyte will land somewhere and grow into a mature 2N structure that we think of as laminaria, or we think of as kelp or seaweed. But that species of seaweed has a lot of different life stages that you might not have known about. And those life stages go in and out from 1N to 2N tissues. You can see here the male and female gametophytes. Remember, those are 1N multicellular tissues under a microscope. And you can see that the male gametophytes are kind of more fringy, and they look like little, almost like green snowflakes or something. And then the female gametophytes have more of a solid structure to them. So there is some dimorphism, some difference between the male and female gametophytes. But those are all haploid cells. Those all have the same number of chromosomes as an egg or sperm cell. The only difference is that these are descended from spores, so they are 1N. We call that process sporic meiosis. So that is, meiosis produces spores, not gametes. We undergo gametic meiosis. That is, when we undergo meiosis, the result is gametes, right? Sperm and egg. Now let's revisit our friend the volvox. Now remember, the chlorophytes were in a lab, and we looked at these as colonial organisms. That is, colonies of somewhat independent cells that grow together in a circle and are kind of like a proto-multicellular organism where they behave as one, but they can also move around individually if they're broken up and continue to grow. So not only do they have an asexual cycle, and this is illustrated on the top right, you can see that the gonidium reproduces daughter colonies. So undergoing mitosis, they can produce and bud off these little daughter colonies of duplicated tissue. It actually spends a lot of its life cycle in a one-end state. When it does create eggs and sperm, they combine to make a zygote. But that zygote immediately differentiates through meiosis into one-end tissues. Volvox essentially spends most of its time, most of its life cycles, and even a majority of its reproductive cycles in a one-end state. It really is only 2N, it's only diploid, during a brief moment after fertilization when a zygote, a 2N zygote, is created. But that zygote almost immediately undergoes meiosis and creates 4 haploid embryonic cells that then begin to duplicate and create that synobium, that orb of tissue that we think of as the volvox, which is actually a colony of one-end cells. We call this zygotic meiosis, and that's meiosis directly from the zygote that immediately produces one-end tissues. Let's look at the asexual cycle of volvox. You can see that inside these little regions within the colony of volvox that makes a little sphere, there are these little areas, these little spaces where you have gonidium. Those gonidium contain cells that will reproduce through mitosis and form a sphere of cells. That ring or that sphere with an opening, the phylopore, will invert itself and expose the flagellum to the outside. You can see here there is a colony of volvox, or several colonies of volvox, and within them are daughter colonies that have formed through this reproductive cycle. Remember, this is asexual, so this is a lot like cloning. They're just reproducing their one-end tissues and forming these little daughter colonies within the larger colonies. In this species of volvox, you can find daughter colonies growing within the daughter colonies. So how do autotrophs eat? How do they fuel their metabolic systems? They use pigments. We discussed accessory pigments, but we'll review it again here. Remember, just like in our lab, if you smash up a plant or any photosynthetic organism, you'll end up with some kind of colored liquid, and that liquid contains the pigments that are used by the chloroplasts. Well, those pigments are excited by light, and not all light excites all the pigments in the same way. So if a plant wants to more efficiently make use of all the different wavelengths of light, it would help to have a bunch of different pigments that pick up different wavelengths of light. You can imagine that the light coming from the top of a canopy in a rainforest, which is exposed directly to the sun, has a different availability of wavelengths than plants that grow on the forest floor. On the forest floor, a lot of light is filtered from the above canopy, and so it's different. It has different wavelengths available. To deal with that, a lot of plants have evolved different pigments to pick up those different wavelengths. And you can see here, we've split it up into its individual pigments that were contained within that colored liquid that resulted from smashing up those leaves. And when we run acetone up the paper, it breaks them up. Why does that happen? Well, as the pigment runs through the matrix of the paper, the larger molecules will be able to move different distances. So this is kind of a way of dividing up the pigment molecules by their size, and it's a convenient way to see how many different kinds of molecules are within there. In this instance, we have 4 different pigments. We have carotene, we have xanthophyll, we have chlorophyll A, and chlorophyll B. Now here's an example of a bunch of different chromatography runs. But you can see there's a bunch of other pigments, not just chlorophyll A and chlorophyll B. You have beta-carotene, you have zeaxanthin, lutein, vioxanthin, neoxanthin, fucoxanthin. These are all unique pigments that help pick up different wavelengths of light. So while plants generally appear to be green, they do have different shades, and that is the result of a different palette of pigments to more efficiently pick up the wavelengths at the location and the conditions that that plant evolved in. We looked at this graph in our lecture before, but I'll remind you that when we see a color reflecting off of a plant, that actually shows you the pigments that the plant is not using. Those are the wavelengths of light that it's rejecting. That being said, if a plant has a lot of different pigments, so if all of these pigments in this example were coming from one plant, it would essentially absorb a lot of those wavelengths, and what you would see would be those valleys, those troughs that are within the visible light spectrum. In this case, this plant or this photosynthetic organism has a trough in its pigments, that's a valley, right around 500 nanometers, and that happens to be green. So this organism, while it's using 5 different pigments to photosynthesize, it essentially looks like a green organism, and that's because that's the wavelength that it's not using. Now, what about all those troughs on the outside, below and above all those peaks? Well, that's outside of the visible spectrum, so we can't see those wavelengths very well. Looking at bacteriophytes and diatoms from space, you can see that their greenish blue color is suggestive of maybe a different suite of pigments than those plants growing on the shoreline. So there's a big island there with a bunch of conifers probably, but just offshore you can see these diatoms, which are autotrophs, are photosynthesizing using different pigments, and those colors, instead of looking just green, have a bluish color to them. Well, that's a great segue into talking about a new group of protists, the diatoms. Diatoms also make shells, and we call shells tests as well. This species is called Pseudonychia, and I like this example because this diatom produces what's known as domoic acid. Domoic acid is a toxin, and it's naturally present in a lot of different organisms that are unicellular or microorganisms that grow offshore. Domoic acid bioaccumulates in the tissues of predators. So in this example, you can see that those producers, those little diatoms in the bottom, as they're eaten by zooplankton, the parts per million of domoic acid increases in the tissues of the zooplankton. Those zooplankton that have eaten these diatoms are a little bit toxic. Well, the small fish that eat those zooplankton, they're now ingesting a slightly higher level of domoic acid, and as a result, they become a little more toxic. So they have an even higher concentration, a higher parts per million of domoic acid in their tissues. Well, those small fish are then eaten by larger fish, and the parts per million of domoic acid in their tissues goes up even more. Finally, you have organisms like birds or seals or larger organisms that eat those larger fish, and they suffer the most severe consequences of domoic acid because it's accumulated at such a high concentration in the tissues of those fish. The results are these mass die-off events, but here you can see a sea lion that's suffering the consequences of ingesting fish that have that toxin, domoic acid. To the right is a pelican that has also ingested fish that have domoic acid. There's an anecdotal example that is kind of interesting, and it kind of ties in popular media with our conversation on domoic acid. Here's an example of animal weirdness from NBC News where a drunk pelican hit a windshield. Well, I don't think that pelican was drunk. I think that pelican was suffering from the neurological effects of domoic acid. It probably ate some fish that had really high concentrations of domoic acid in it. And this happens whenever you have a red tide. You'll have a lot of different wildlife acting kind of strange because that domoic acid affects the behavior and the neurological system of vertebrates. Here in 1961, you can see in the Santa Cruz Sentinel there's a big one-pager talking about the seabird invasion, where a bunch of seabirds came in and started hitting homes and running into things. Well, all those seabirds were suffering from domoic acid, produced from diatoms like Pseudonetia. That was such a big event, it kind of ingrained in the minds of a lot of people. And not shortly after, we have the film The Birds from Alfred Hitchcock, which showed a bunch of birds acting crazy. Well, that film stemmed from this seabird invasion in Capitola near Santa Cruz, where a bunch of birds were affected by domoic acid and started running into windows and acting pretty wild. Well, as a result, a lot of people now have phobias of birds, and a lot of people don't like birds because of this movie. So it's kind of an interesting story that shows that our phobias and fears can stem from protists. Now let's talk about the phaeophytes. And for an example, we'll talk about fucus. Now, fucus is something you might typically call a seaweed. It's like a kelp. Superficially, seaweeds and kelps, they look a lot like plants, but they're not actually plants. They're protists. And a lot of the reasons why they look similar to plants is convergent evolution. To solve the same problems in their environment, they've evolved similar structures, but those structures actually don't share a common ancestor. So the blades, or what you would think of as leaves on a fucus, don't share any genetic similarity to the blades of a tree or a plant, but they serve the same function. They serve as a surface to increase the area that plant can undergo photosynthesis and collect light energy. One problem that plants have on land is how to grow taller. They have to form stronger structures, and they have to resist wind and things like that. Well, underwater, you don't have to worry about strength so much as the ability to stay closer to the sunlight. Well, to solve that, a lot of seaweeds include what's called a float, and that's a little kind of a gas-filled structure that keeps those blades afloat near the surface of the water where there's the most light. Below the float, you have the stipe, which is analogous to a stem in a plant, but remember, has a slightly different purpose here. Instead of growing tall, the stipe is more used to keep the float and keep the blades within a certain area. Below the stipe, you have what's known as a holdfast. A holdfast is used simply to do that, to hold the plant fast onto rocks, to hold the plant in place and anchor it so that it doesn't float away. Remember, a lot of these organisms live in a very specific area in the water column, and if they were to drift away, they wouldn't do very well. Phaeophytes are like the brown algae, and this example is Macrocystis porifera. That's giant kelp. You can see that these grow huge. Now, while we were just talking about single-celled organisms one lecture ago, we've immediately moved on to some of the largest organisms in this ecosystem. Kelp are so large, they're foundational in a lot of the life histories of animals that live within their habitat. One organism you're probably familiar with that relies on Macrocystis is the sea otter. Remember, sea otters eat sea urchins, which are predators of Macrocystis. And this sea otter has one of those sea urchins on its belly. Something you might not know about sea otters is they eat so many purple sea urchins that their bones actually appear purple. And in this case, the teeth of the sea otter actually grow purple. Sheephead are another species that are tightly associated with Macrocystis. And you might be familiar with sheephead because they have kind of interesting teeth. And one of our state ambassador organisms is this, the Garibaldi. Garibaldis live amongst the Macrocystis forest off the California coast. Lastly, we'll talk about rhodophytes, the red algae. There are many types of red algae. Red algae occur in such a diversity of morphologies that we divide them up into several groups based on the way they grow. So these are coralline algae, mesophyllum, and these have calcium carbonate structures. Now remember, calcium carbonate is something that's only biologically available at really specific pHs, so this is another species that might be negatively affected by ocean acidification. Some red algae are used in cooking, and they can create kind of a gelatinous effect, which is, of course, not animal-based. A lot of vegan jellos use different red algae to give that effect. Red algae are a very, very old lineage, and some of them haven't changed almost at all. Here we have an example of a fossil from 1.2 billion years ago, and this is Bangiomorpha pubescens. Well, here is a Bangia algae today, and you can see that superficially or morphologically, they appear almost indistinguishable. Lastly, I'll leave you with these facts. What do you think the percent of oxygen produced by algae is? Well, it's 30 to 50% of our oxygen. How much red algae extract do you think the average American consumes daily? Well, it's 250 milligrams. How long do you think kelp get? 200 feet. How many times more productive is algae than traditional crops as biofuel? 50 times more. What's the number of moderate to large algal cells per milliliter of water needed for an algal bloom? Remember, like the red tides. 15,000 cells per milliliter. How many cells do most basic algae have? That's 1. That's our discussion on autotrophic protists. Now it's time for us to move on to a new group of organisms, and that group of organisms is the plants. So, our next lecture, we will be talking about the plants.